Hi, I'm Nina Hunter. Thank you to the Alliance for Regenerative Medicine for the opportunity to speak today and to provide you with some of our corporate updates. Here's just a slide of our forward-looking statements for your reference. I will be making forward-looking statements today in my presentation. Regenxbio is a company of now about 400 full-time employees with the mission of seeking to improve lives through the curative potential of gene therapy. We have a leading pipeline of AAV therapeutics with the potential to deliver one-time treatments. We announced last year in 2021 a strategic partnership with AbbVie to develop and commercialize AAV therapeutics for retinal diseases. We have proprietary AAV therapeutic manufacturing with analytics, delivery device, and global supply platform in-house. We have a strong balance sheet and a 5x25 strategy to progress 5 AAV therapeutics from our internal pipeline or license programs into pivotal stage or commercial products by 2025. RGNX's NAV technology platform is a proprietary AAV gene delivery platform consisting of exclusive rights to over 100 novel AAV vectors, including AAV7, AAV8, AAV9, and AAVrh10. Our technology platform is a significant advancement over earlier generations of AAV vectors in delivering genes into cells with differentiated attributes. These unique benefits have been demonstrated in clinical trials in various animal models. We and our third-party NAV technology platform licensees are applying this technology in the development of a broad pipeline of candidates, including late-stage and commercial programs in various therapeutic areas. We believe that these programs together further validate the NAV platform and provide additional data that collectively drive forward the advancement in the AAV gene therapy field. Here is a snapshot of our internal pipeline that is broad and can be divided into 3 categories. We have retinal, neuromuscular, and neurodegenerative diseases. We have 2 clinical stage programs, RGX-314 for wet age-related macular degeneration, or wet AMD, by subretinal delivery, and RGX-121 for mucopolysaccharidosis type 2, MPS II, or Hunter syndrome. So today, I plan to provide a brief overview of our programs across these indications, starting with retinal diseases. So as I mentioned, in 2021, we entered into a collaboration and license agreement with AbbVie to develop and commercialize RGX-314, a potential one-time gene therapy for the treatment of wet AMD, diabetic retinopathy, and other chronic retinal diseases. Wet AMD is characterized by loss of vision due to excessive blood formation between 2 layers of cells in the retina. This excess blood formation results in fluid leakage that leads to physical changes in the retina and vision changes. As this process becomes more severe, blindness can result from scar formation due to hemorrhaging. RGX-314 consists of the NAV AAV8 vector, which encodes an antibody fragment designed to inhibit VEGF. RGX-314 is believed to inhibit the VEGF pathway by which new leaky blood formation grows and contributes to the accumulation of fluid in the retina. And currently, we are advancing programs in 2 separate routes of administration for RGX-314 in the eye, through a standardized subretinal delivery procedure, as well as through a suprachoroidal space delivery. We have 2 pivotal phase trials with subretinal delivery and 2 phase 2 trials with suprachoroidal delivery. The suprachoroidal delivery path allows for an in-office, non-surgical procedure. Atmosphere and Ascent and AV8 are trials for wet AMD, and Altitude is for diabetic retinopathy. We just recently shared some very exciting data updates from some of these trials, highlighting the potential impact of RGX-314 for the millions of patients facing vision loss. Our trials for both wet AMD and diabetic retinopathy, 2 prevalent conditions, have enrolled hundreds of patients and dosed hundreds of patients, and I think it's the most number of patients ever dosed in clinical trials using AAV gene therapy. I won't go into the details today, but we've demonstrated that RGX-314 continues to be well tolerated and has long-term durable treatment effects of up to 4 years via subretinal delivery. Pivotal trials are expected to support a BLA submission in 2024, and we look forward to providing more updates in the near future. So now let me tell you a bit about our neuromuscular program in Duchenne muscular dystrophy with our product candidate RGX-202. Duchenne muscular dystrophy is caused by mutations in the DMD gene, which encodes dystrophin. DMD gene mutations result in alterations of the structure or function of dystrophin or prevents any functional dystrophin from being made at all. The lack of dystrophin with repeated muscle, contraction, and relaxation leads to damaged fibers that weaken and die over time, resulting in progressive muscle weakness, including cardiac dysfunction. Our approach is to restore function by delivering microdystrophin to act as dystrophin using AAV8, and with a microdystrophin construct that contains functional elements of the C-terminal domain, known to recruit several key proteins to the muscle cell membrane with the goal of providing additional potential benefits that are unique. We are preparing for the initiation of our Affinity Duchenne Phase 1/2 trial, including readying clinical trial sites and manufacturing additional clinical supply for the trial. We anticipate dosing the first patient in the first half of 2023. And now switching to an overview of our neurodegenerative programs, we have 3 programs for monogenic gene replacement using AAV9 delivered intracisternally for rare pediatric lysosomal storage diseases, so MPS II, also MPS I, Hurler syndrome, and as well as CLN2, also known as Batten disease. Because these candidates are delivered intracisternally directly into the central nervous system, they're designed to address the extreme high unmet need of these children with neurological manifestation of their diseases. So let me just walk you through one of these programs, RGX-121, in more detail. It is designed to deliver the gene that encodes iduronate-2 solvatase, or I2S enzyme, using the AAV9 vector. MPS II is a rare X-linked recessive disease caused by deficiency of I2S2, leading to an accumulation of glycosaminoglycans, or GAGs, such as heparan sulfate in tissues, which ultimately result in cell, tissue, and organ dysfunction, including in the central nervous system. In our Phase 1/2 trial, we demonstrated in 14 patients dosed across 3 cohorts, so 3 different dose levels, no severe adverse events related to the drug. We observed dose-dependent reductions in biomarkers in the cerebrospinal fluid across these cohorts. And in cohort 3, where patients received our highest dose, a component of heparan sulfate called D2S6 closely correlated with severe MPS II disease approached normal levels at 48 weeks. This is important because heparan sulfate and its component D2S6 are GAGs that are key biomarkers in I2S enzyme activity. And buildup of GAGs in the cerebrospinal fluid MPS II patients correlates with clinical manifestations, including neuronal development deficits. And in these patients, we demonstrated improvements in neurodevelopmental function and caregiver-reported outcome in cohorts 1 and 2 up to 2 years after administration. So just briefly here, as you can see, heparan sulfate measured in cerebrospinal fluid across the 3 cohorts at week 48 and at 2 years on your left-hand side. And then at week 48, median reduction of CSF from baseline around 34%, 53%, and 63% in cohorts 1, 2, and 3 respectively. And D2S6 disaccharide demonstrating at week 48 levels continuing to show dose-dependent reductions across cohorts, with cohort 3 approaching normal levels. A median reduction from baseline around 32%, 70%, and 83% in cohorts 1, 2, and 3, respectively, is seen at week 48. And the majority of patients in all 3 cohorts demonstrating decreased levels at the last time point that was available. And with these encouraging biomarker data, we have importantly also demonstrated that RGX-121 has a positive impact on neurodevelopmental function as measured by cognition, expressive language, and fine motor skills. And in fact, what these graphs show is that patients with cognitive function above a -2 standard deviation at baseline are those that may benefit the most from our potential candidate. MPS II is a progressive neurodegenerative disease, which is why I believe it's important that these children have access to this potential treatment as early as possible. So taking these data together, we have recently announced our pivotal program campsite, including a phase 3 trial that is active and enrolling up to 10 patients to support a BLA filing in 2024 using the accelerated approval pathway. GAGs and CSF have the potential to be considered a surrogate endpoint that is reasonably likely to predict clinical benefit. We will be using our commercial scale cGMP material from our proprietary high-yielding suspension-based manufacturing process called NAV Express, and we look forward to advancing our trial as quickly as possible with the aim of providing a much-needed treatment option for the MPS II community. And lastly, I'll end here with a little more about NAV Express that we are currently making in our brand new state-of-the-art gene therapy manufacturing innovation center at our headquarters in Rockville, Maryland. Just opened this summer, we believe our in-house manufacturing capabilities will enable us to rapidly transition production processes across the product lifecycle and efficiently advance AAV therapeutics from research and early development to clinical trial programs and commercial readiness, and ultimately to patients who may benefit from these potential one-time treatments. Bringing our manufacturing in-house allows us to control the process from the beginning to the end and the flexibility to support a wide range of clinical and commercial needs, all while ensuring quality for the patients. And at the foundation of all of this, of course, are amazing teams of full-time employees across disciplines and functions working together to advance our programs for patients in need. So with that, we look forward to providing more updates in the future, and thank you for your attention today.